Hello, and thank you for tuning in to my poster presentation. I'm Jennifer Hellal from BRGM, and I will present you some work on bacterial biotransformation of Chlordecone and two degradation products from the InSitu Chemical Reduction process, ISCR. This is work carried out by colleagues of mine as well as colleagues from the UMR Genoscope. Chlordecone is an insecticide that was extensively used in the French West Indies in the 70s and 80s in banana plantation. ISCR is a promising process that enables to reduce chlordecone in soils and form hydrochlordecones, which have been shown to be less toxic than chlordecone. So our question today is, we know that chlordecone can be degraded, but can these metabolites also be degraded by the same consortia? Our experimental setup consisted first of enriching a chlordecone degrading consortia over several months, and then inoculating this consortia either with chlordecone or with two hydrochlordecones, one that had lost one chloride or one that had lost 3 chloride. So briefly, some results. Here in the graph below you have the evolution of chlordecone in the sterile and in biotic conditions, and we can clearly see a decrease in the chlordecone concentrations over time, and at the same time, with the pink line, we can see an increase in a C9 molecule, which has been recognized and found before to be a direct product issue from bacterial degradation of chlordecone. In the other conditions, with the hydrochlordecones, we also saw a decrease of these molecules over time during the incubations, but there was no immediate obvious metabolite formed, and certainly no C9 molecule. So with the colleagues from the UMR Genoscope, we went looking for new formed metabolites and found that the hydrochlordecones had actually been functionalized and had a sulfide added to their structure, which meant they were no longer detectable with the previous method. We also looked at bacterial diversity in the incubations over time. Here you have the results from high throughput sequencing at the end of the incubations. We can see that in the chlordecone condition we had a diversity that was comparable to the inoculum, with the presence of disulfovibrio, which has previously been shown to degrade chlordecone, whereas in the other conditions diversity evolved, and it's other genus that now dominate that are not known for chlordecone degradation. So in conclusion, we found that both chlordecone and two of the metabolites issued from ISCR could be biotransformed with an enriched community, that the metabolites formed are different depending on the added molecule, possibly due to different metabolisms and pathways. We found neoformed molecules with a functional sulfur group, and we also showed that bacterial diversity was modified according to the incubation conditions over time, with changes in dominant bacteria, possibly due to more tolerant conditions with the hydrochlordecones rather than with the chlordecone itself. Thank you very much. Don't hesitate to get back to me if you have any questions.